You can share the screen now, sir.
Okay, just wait.
Okay, okay, sir.
Will we start? I mean, should I start?
Yes, sir, we can start.
Just a moment, please. No. So, yes, good evening, everyone. This is Dr. Abhijeet, and welcome to yet another session of Lipid Metabolism Part 2. So, the Oracle, came up with exam mastery live classes that has already started. Right, in this particular session, we are revising the all important questions that can be asked in the upcoming university exams that will be no that would be held in August. Apart from that, guys, all the volatile topics that I feel which is very, very important, I will be doing quickly revising it. Okay, all the conceptual part will be discussed over here, right? And even the most important or potent questions for the upcoming exam will be discussed, right? Apart from that, apart from the university exams, it is most no essential for the NEET PG and FMG exam as well. So, you already know we are going to discuss the lipid metabolism part 2 today, where I will be discussing about the fatty acid oxidation, right? Apart from that, I will be discussing certain diseases today. If I get time, I'll be discussing lipogenesis, ketogenesis, and ketosis also. Otherwise, the some parts we'll see in the next part, right? Now, guys, about the Oracle app, you already know we have the live classes, recorded lectures. Apart from that, the notes are curated in such a way that all like it's very, very important, right? Apart from that, after every topic, there are certain MCQs, right? They are clinical based. All the previous year questions, all the previous year topics are being covered, right? So there is a plan which you can see on the screen. And apart from that, if you want some additional discount, you can use my code Dr. Abhijeet 20 for the subscription, right? So even you can book a slot for no personalized counseling session. So without wasting much more time, let us start with the lipid metabolism part 2. So here is our first question, guys. Write in detail, and it's a 10 marks question. Obviously, it will be a 10 marks question, right? Now they are asking, and I will be discussing in my own way first, right? Then you can refer to the notes which is given in the Oracle app, right? So the first thing, it is about the beta oxidation or the fatty acid oxidation, and we have to calculate the energy, the bioenergetics of palmitic acid. Now, let us start. We will be discussing it under four headings, guys. Right? We will be discussing it under four headings. Firstly, I will be discussing the concept of, yes, the concept. Now, the very first thing in the concept, guys. Now, why fatty acid oxidation is necessary? Why this acid oxidation is required in our body? Now, you guys already know. Yeah, let us just see if it is 1 to 4 hours after food. 1 to 4 hours after food, you already know this state. This stage it is called as the well-fed state, right? This stage it is called as the well-fed stage, right? Now in this stage, what is happening, right? Now, these are the stages of fasting that I'm discussing. These are the stages of fasting. You have just consumed the glucose. What will happen? Yes, glycolysis will take place, energy will be there, and yes, glycogenesis will occur. Glycogenesis will occur. Apart from that, in the second stage, where we say it is the early fasting, and it is somewhat between 4 to 16 hours, right? And it also depends on the body weight of the person, right? Also depends upon the amount of meal consumed. So it actually can vary. These hours can vary, but an estimate that I'm giving you over here, it is the 4 to 16 hours. And you already know whatever glycogenesis has been occurred, whatever the glycogen was stored, right? Now what will happen? You're not consuming any food in 4 to 16 hours. Whatever the glycogen has been stored in the liver, this will break down. And this is called as glycogenolysis. This is called as glycogenolysis, right? The glycogen will be broken down to form glucose. Apart from that, guys, now let's come to the next stage, that is 16 to 48 hours after food. And you already know this is called as the fasting stage, actual fasting, 16 to 48 hours. Now, in this case, all the glycogen has been depleted. Now, what will happen? There is something called as gluconeogenesis, which we have already discussed in the carbohydrate metabolism. New glucose will be synthesized from different sources. From pyruvate we have discussed, right? From glycerol we have discussed. From lactic acid we have discussed. So glucose will be synthesized using some other sources. For example, amino acid. We have already discussed this. Now comes a stage where the fasting is more than 2 days. Where the fasting is more than 2 days. Now, guys, or it can be up till 5 days without food. What happens in this case, guys? What happens in this case? It is called as prolonged fasting. It is called as prolonged fasting, and this is something very important. And this is something called as starvation. 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 Now, in this stage, guys, where the fasting it is from 2 days up till 5 days, what will happen, guys, now? There is no glucose, there are no other sources, there is no amino acid left, there is no lactate left, there is no acetyl coenzyme A left. Now, over here, what will happen? There is something called as fatty acid oxidation will take place. And this is something that you have to remember. Fatty acid oxidation will take place. Plus, there will be ketone body synthesis. I'll be coming to this ketone body a bit later in the lecture. But remember, there is something called as fatty acid oxidation will take place in the body. And this is nothing but the beta oxidation of fatty acids. Beta oxidation of fatty acids. And this is the stage which will provide ATP, right? Which will provide ATP to the body. So that is something you have to remember, the concept why the fatty acid oxidation is necessary in our body. Obviously, in cases of prolonged fasting, and this is something that you have to remember when you are without food for 2 to 5 days, and it is called as prolonged fasting or starvation. Beta oxidation of fatty acid will occur, and that will result in the formation of ATP. What is the process? We'll be discussing it right now, right? So that is something you guys have to remember. Now. Now, there is something called as if the fasting is for more than 5 days, then there is something called as prolonged starvation. Prolonged starvation, but that is yeah. So not our point of concern over here. So we'll be focusing on this fourth wala point, that is the fatty acid oxidation. Fatty acid oxidation. Now, now over here I will be discussing the beta oxidation of saturated fatty acid as given in the question. It is saturated fatty acid, and you already know which acid I'm talking about. It is the palmitic acid. It is the palmitic acid. It is the palmitic acid, right? Now, firstly, guys, you just tell me what is the general formula for fatty acid. General formula of fatty acid. Now, you guys already know it is RCOOH, right? R, it is a long carbohydrate chain, right? R, it is nothing but the long hydrocarbon chain. Long hydrocarbon chain. I've already told you it can be CH3, CH2, CH2, CH2. So it can continue. So this is a long hydrocarbon, hydrogen and carbon chain. And you know the general formula for fatty acid is this. Now, now, two questions that is important over here. One, it is the most common fatty acid that undergoes beta oxidation, and that is nothing but, yes, the palmitic acid. The palmitic acid. And palmitic acid is having 16 carbons, guys, right? It is having 16 carbons. And remember, yes, one more thing, that is most common fatty acid oxidation. Most common fatty acid oxidation, it is beta oxidation. It is beta oxidation. Now, guys, there are more no fatty acid oxidation, guys. You should also know that there is oxidation of very long chain fatty acids, right? There is oxidation of unsaturated fatty acids. There is oxidation of odd chain fatty acids. So we have multiple no types of fatty acid oxidation. We have different types of fatty acid oxidation. As per our question, I will be discussing on the beta um, oxidation of the fatty acids, right? So there is also called as the minor pathways of oxidation, that is alpha oxidation and omega oxidation of fatty acids. So we are not concerned about that. We'll be discussing about the beta oxidation. Now, guys, apart from that, you have to remember what are the sites of beta oxidation of fatty acids. Beta oxidation of fatty acids. Now, what are the sites, guys, where this fatty acid oxidation, yeah, beta oxidation of fatty acids, right? Now, where does this take place? Guys, you already know we have three important sites over here. You should remember the first site, it is the liver. The second, it is the adipose tissue, right? It is the adipose tissue. The third one is the muscle. The third one, it is the muscle. We have liver, adipose tissue, and muscle. We have liver, adipose tissue, and muscle. Now, apart from that, there is one more question that they will ask, which organelle? Yeah, which organelle over here? It is the mitochondria. Mitochondria, right? It is the mitochondria. So sites you should remember the organelle, which part of the cell organelle it is, mitochondria. The different organs you should remember. Apart from that, you should remember the beta oxidation. Now coming on to the steps of beta oxidation. We have to discuss the steps, right, in detail. So let us just try to focus on the steps of the beta oxidation. Steps of beta oxidation of fatty acids. Steps of beta oxidation of fatty acids. Now, guys, let us just start one by one. What are the three basic steps? There are three basic steps for beta oxidation. There are three basic steps for beta oxidation. The very first one, guys, it is activation of the fatty acid. It is activation of fatty acid, right? We have transport of activated fatty acid to mitochondria. And the third one, it is the beta oxidation. It is the beta oxidation. Activation of fatty acid, transport of activated fatty acid to mitochondria, and beta oxidation. Right? Just a moment, someone is calling. Now, coming on to the very first one, guys, that is the activation of the fatty acid. Activation of fatty acid. Activation of the fatty acid. Now, let us just start. Now, you guys already know we have a substrate over here, and that substrate, it is nothing but the fatty acid. That substrate, it is nothing but the fatty acid. Right? Now, this fatty acid is nothing but this component, RCOOH. Now, this fatty acid will be changing into, yes, acyl coenzyme A. Acyl coenzyme A. And this is nothing but a fatty acid residue. Fatty acid residue. Remember, guys, in the activation part, always the energy will be utilized. Always the energy will be utilized. One ATP will be broken down into one AMP, right? And the enzyme over here, it is acyl coenzyme A synthase. And that is something called as the thiokinase. The thiokinase. Right? Kinase, guys. I've already told you, right? Kinase means basically shift of two phosphates group are being shifted from ATP molecule to this fatty acid. And this is something called as the acetyl coenzyme A. Acetyl coenzyme A, right? Acyl coenzyme A, right? So acyl coenzyme A will have many carbons, right? It will have many carbons. Now, over here, guys, you have to remember this step. This step requires two high energy phosphates. Two high energy phosphate. Obviously, ATP is converting into AMP, right? Tri is converting into monophosphate. Obviously, there are two high energy phosphates are lost, right? Now, the enzyme over here, this kinase basically, yeah, it belongs to ligase. The enzyme belongs to which group? The enzyme belongs to ligase. And you already know, guys, this ligase is something which needs ATP, right? It is the only energy requiring step in the, oxidation pathway, right? Requiring step in fatty acid oxidation. Fatty acid oxidation. Now, this activation is somewhere, it takes place in the cytoplasm, guys. It takes place in the cytoplasm. It is not in the mitochondria. If you want to mention, it occurs in both. No, it occurs in both, like you can say, this cytoplasm and mitochondria both. So yes, I have written mitochondria, but you can mention cytoplasm also because activation parts take place in the, yeah, this cytoplasm. Only energy requiring step in the fatty acid oxidation where energy is too high, phosphate molecules are being required, right? Now, apart from that, now this enzyme, okay, this enzyme, it is basically located in the outer mitochondrial membrane. You guys already know that mitochondria has two membranes, right? The outer mitochondrial membrane and the inner mitochondrial membrane. So this enzyme is basically located on the outer mitochondrial membrane, and this activation is basically taking place in the cytoplasm, right? So this is the first step that you have to remember. Right? So yes, that is the activation of the fatty acid. Now coming on to the next part, that is the transport of activated fatty acid to mitochondria. Now guys, the second one, That is the transport of activated fatty acid to mitochondria. Transport of activated fatty acid to mitochondria. Now, when I say activated fatty acid, guys, you already know the product was acyl coenzyme A. Acyl coenzyme A. It has many carbons, right? Acyl coenzyme A. Now, over here, you require a transporter, guys. Over here, you require a transporter, okay, for transporting this activated fatty acid to mitochondria. Now, let us just, I'll draw the diagram also, guys. You already know that this is, okay. Let me just first write down the parts and then I'll draw a diagram for you people. So the transporter basically here required is CAT. The transporter is carnitine. Carnitine. Now, what is this carnitine, guys? What is this carnitine? Carnitine is beta hydroxy gamma trimethyl ammonium butyrate. Ammonium butyrate. So it is beta hydroxy gamma trimethyl ammonium butyrate. Okay. Now this carnitine, remember beta, it is synthesized in the muscle. It is synthesized in the muscle. These are very, very important points that you have to know, pen down in the exam as well. Right? Now, for the synthesis of the carnitine, guys, basically two raw materials are required. Okay, for the synthesis of carnitine, two raw materials are required. That is again a very important question for the Viva part. That is the two amino acids. Two amino acids, and you should remember it is lysine and methionine. It is lysine and methionine. It is lysine and methionine, right? So that is something you have to remember. Okay. So now coming on to the next part, guys, coming on to the next part, right? Now, there is something very, very important part that fatty acids. Now, again, this part is something very important that you have to remember. The fatty acids with carbon atom less than, with carbon atoms less than 14 does not need carnitine. Does not need carnitine, right? Obviously, but over here we require carnitine because palmitic acid is having how many carbons, guys? It is having 16 carbons, right? It is having 16 carbons, right? Now, let us just try to see what is happening. What is happening, guys, over here? Okay, you already know we have the outer mitochondrial membrane. Let us just write it as the outer mitochondrial membrane. Okay, and inner to that, you already know on the outer mitochondrial membrane, we have an enzyme that is located, right? And that is called as CAT1. That is CAT1. 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 Okay. Now, what is this CAT1, guys? First, just try to remember this CAT1, it is nothing but the carnitine acyl transferase. Carnitine acyl transferase 1, or it is also called as CPT1, right? Or it is also called as CPT1, carnitine palmitoyl transferase 1. Carnitine palmitoyl transferase 1, right? P is palmitoyl, right? Palmitoyl transferase 1. Palmitic acid ke liye, we are discussing about the palmitic acid wala part, right? So, yes. Now this acetyl coenzyme A, okay, we have something called as the inner mitochondrial membrane also, right? We have the inner mitochondrial membrane. mitochondrial membrane. And yes, inside you already know we have the mitochondrial matrix. We have this mitochondrial matrix. Yes, we have two. The mitochondria has two membranes, the outer mitochondrial membrane, the inner mitochondrial membrane, and inside it is the mitochondrial matrix. Mitochondrial matrix. Now, what is happening? This acyl coenzyme A, let me just write it with this. Yes, this acyl coenzyme A will now come into this between the outer and the inner mitochondrial membrane. This is acyl coenzyme A plus carnitine. Okay, it is plus carnitine. Now, what it will form, guys? It will form acyl carnitine plus coenzyme A. Acyl carnitine plus coenzyme A. Now, guys, this acyl carnitine will now try to go inside the mitochondrial matrix. Okay, inside the mitochondrial matrix, and that is by the help of an enzyme over here that is called as translocase. Translocase. Let me write it more clearly, guys, so that it is, yeah, I'm just writing it translocase, right? Just put a box. So acyl carnitine will come over here in this mitochondrial matrix now. Acyl carnitine. Let me write it with blue. Yes, acyl carnitine. Now, in the mitochondrial matrix, it will again combine with acyl coenzyme A, right? It will again combine with acyl coenzyme A, right? And yes, what will happen? What will happen now, guys? This acyl coenzyme A will now they will give a product that is acyl carnitine plus coenzyme A will be there, right? Now, what will happen? There is again another compound over here that is called as, yes, the CAT2. The CAT2. Okay, that is nothing but the carnitine acyl transferase 2 or carnitine palmitoyl transferase 2. Right now, what it will do? It will again, yes, we already know we have this, yes, acyl carnitine plus coenzyme A, right? Now, what will happen? This will again change into this carnitine plus acyl coenzyme A. Okay, and this carnitine will again go outside into the outer mitochondrial membrane, I mean to the cytoplasm part, right? So that is something you have to remember. So it is nothing but, guys, you just have to remember acyl coenzyme A plus carnitine, okay, will be forming acyl carnitine plus coenzyme A. This acyl carnitine will go into the mitochondrial matrix, okay, that is acyl carnitine. It will again combine with this coenzyme A, right? So what will happen? There will be acyl coenzyme A which will be formed and carnitine will be released. This carnitine will go again back to the cytoplasm or the outer mitochondrial membrane. That is what I have mentioned over here, right? Now, coming on to the third step, that is the most important, and that is called as the beta oxidation proper, right? That is called as the beta oxidation proper. And remember, guys, this is something which occurs in the mitochondrial matrix. That's why it occurs in the mitochondrial matrix. Mitochondrial matrix. Now, you already know we have acyl coenzyme A, right? Now, this acyl coenzyme A will now transfer into trans-enoyl coenzyme A, right? It will convert into trans-enoyl coenzyme A. And to this step, basically an enzyme will be used, right? The flavin adenine dinucleotide. FAD will change into FADH2. Now, from trans-enoyl coenzyme A, what will happen? Hydration will take place. Now, from acyl coenzyme A to trans-enoyl coenzyme A, what is taking place? Oxidation. Oxidation, right? Oxidation, basically the hydrogen is getting removed, right? So it is the oxidation part. Now this hydrogen which is removed, it is accepted by the FAD. FADH2, again there will be release of ATP, right? Now this is trans-phenylyl coenzyme A. Again, what will be added over here? Water will be added over here, right? And this is something called as hydration. And what it will result in? It will result in the formation of beta hydroxy acyl coenzyme A. Acyl coenzyme A, right? Now, acyl coenzyme A, you already know this is all 16 carbon. This is all 16 carbon. Now, beta hydroxyl acyl coenzyme A will convert into beta ketoacyl coenzyme A. Okay, and it's again oxidation, guys, right? Oxidation, or I can simply say the dehydrogenase. The dehydrogenase, that is hydroxyacyl, yes, hydroxyacyl coenzyme A, dehydrogenase, right? Dehydrogenase, you already know whenever we are using dehydrogenase, what is happening? This NAD plus will be converted into NADH. NADH, obviously the energy will be released now. Now this ketoacyl coenzyme A or beta ketoacyl, yes, yeah. So this beta hydroxyl acetyl coenzyme A, it will be converting into beta ketoacyl coenzyme A, which will again break down into two parts: acyl coenzyme A and acetyl coenzyme A. Acetyl coenzyme A is the 2-carbon compound, and acyl coenzyme A, it is a 4-carbon compound. It is a 4-carbon compound. Now, this acetyl coenzyme A, which is released, it will now enter into the TCA cycle. It will enter into TCA cycle. And you guys already know in one TCA cycle, we have how many net gain of ATP? It is 10, right? So that is something you have to remember over here. Over here, let us just try to calculate the energetics. Now, this is how you have to remember, right? Firstly, the activation, the transport, and the beta oxidation proper. Now, coming on to the energetics wala part. Over here, I don't have space, so I'll write it over here. Let us first discuss the energetics of beta oxidation. Energetics of beta oxidation. Now, guys, you already know the question says palmitic acid. Palmitic acid. Now, palmitic acid has 16 carbon atoms. It has 16 carbon atoms, right? Now, when we say number of beta oxidation, guys, you should remember it is 7, right? It is 7. Number of beta oxidation, it will be 7, guys. It will be 7. Now, now how to calculate the number of beta oxidation? You already know that it is number of carbon by 2 minus 1, right? So number of carbon, it is 16 by 2, that is 8 minus 1, that is 7. Number of beta oxidation is 7 beta oxidation. Coming on to the number of acetyl coenzyme. So it is how to calculate this beta oxidation. It is number of carbon by 2 minus 1. Number of carbon, it is 16 by 2 minus 1, that is 8 minus 1, that is 7. So number of beta oxidation is 7. Number of acetyl coenzyme A. Acetyl coenzyme A, guys, it is 16 by 2. Okay, that is 8 acetyl coenzyme A. 8 acetyl coenzyme A. Now, guys, again, makes sense. One acetyl coenzyme A gives, when it enters into the TCA cycle, it will give 10 ATPs. 8 acetyl coenzyme A, it will give how many ATPs? 80 ATPs, right? So number of acetyl coenzyme A, how it is calculated? Number of carbon atoms by 2. Number of carbon atoms by 2. So remember, guys, one beta oxidation. Remember, one beta oxidation has 2 dehydrogenases, right? The 2 dehydrogenases, the 2 oxidation that is taking place, 2 reducing equivalents which are formed. One is 1 NADH and 1 FADH2, right? 1 NADH will give 2.5 ATP according to the latest one, and one FADH2 will give 1.5 ATP. 1.5 ATP. Now, guys, again, I will tell you this is the new wala calculation. Books again says 3 and 2 wala concept. I will calculate from that only, right? But let us just focus on the new wala part. So 2.5 plus 1.5, it is 4 ATPs, right? 4 ATPs. One acetyl coenzyme A, when it enters into the TCA cycle or the Krebs cycle, how many ATPs it gives? 10 ATPs. Now, how many acetyl coenzyme A is formed? 8. 8 acetyl coenzyme A is formed. So you should know palmitic acid. Palmitic acid, it is 7 beta oxidation plus 8 acetyl coenzyme A. 7 beta oxidation means 7 into 4, right? From 1 beta oxidation, we are getting 4. 7 beta oxidations are there. Plus 8, 1 acetyl coenzyme A into 10. Now it is 28 plus 80, that is 108 ATPs. 108 ATPs. Now, guys, again remember, this is the one palmitic acid giving 108 ATPs, but it is the total net gain. It is different. You can very well see in the first step, guys, the 2 ATPs. Okay, let me just write this. 2 phosphate molecules are being used. Minus 2 IP is there, right? So 2 phosphate molecules are used. I've just written in one way. So yeah, ATP converting into AMP, 2 phosphates are lost. So 2 energy compounds are being lost. So 108, okay. So it is, yeah, 108 minus 2, guys. It is 106 ATPs is the net gain. Net gain of ATP. Net gain of ATP. Again, guys, this is this is you already know, right? Now, based on the old calculation, guys, based on the old calculations, based on something which you already know, if one NADH, it is corresponding to 3 ATP, and one FADH2 corresponding to 2 ATP, it is 5 ATPs, right? It is 5 ATPs. You already know if there is 7 beta oxidation, it will be 7 into 5, right? 7 5s are 35 will be there, right? And one acetyl coenzyme A will be giving not 10 ATPs, it will be giving 12 ATPs. Okay, it will be giving 12 ATPs. So 35, right? 35 plus 12 into 8, right? 12 into 8, it is 96. So again, it will be 131. 131 minus 2 again, which was used, it is 129 ATPs. So based on old calculations, you can also calculate. But just follow the new one. If your teacher is saying go for the old calculation, just remember one NADH in the old calculation, it is 3 ATPs, and one FADH2 in the old calculation, it is 2 ATPs. Is that clear, everyone? So yes, let's just try to focus on the next part now. So that was regarding the first part of the question, that is in detail about the fatty acid oxidation, the concept, the site, the parts I've told you, and the 3 steps, and the bioenergetics of palmitic acid, old and the new, both concept I've told you. So whichever your teacher says, just try to follow that, right? Now, sure. Let us now discuss about some diseases. The very first one that I will be discussing, it is the Niemann Pick disease. Niemann Pick disease, guys. You already know the Niemann Pick disease. It is also called as, yeah, the enzyme deficient. It is also called as sphingomyelinase. It is also called as sphingomyelinase, right? Now, let us just try to discuss in my way. Then I will be coming to your way also, right? Niemann Pick disease. Now, Niemann's Pick disease. Now, what happens in this disease, guys? Remember, we have already discussed the types of no the sphingomyelin and all. So remember, beta, the enzyme defective is sphingomyelinase. Sphingomyelinase. Right? Now, what does this enzyme do, guys? This enzyme basically converts the sphingomyelin into ceramide. Right? This is the function of this enzyme. Now imagine this enzyme is not working. What will happen? Again, the concentration of sphingomyelin will be more. Right? Now, what what are the two important findings that you have to remember over here, guys? That is the cherry red spots which will be present. Which will be present. And there will be zebra bodies inclusion. Very, very important. Zebra bodies inclusions, which will be present. Right? Now let us just try to see over here, guys. The enzyme deficient is sphingomyelinase. Progressive neurodegeneration occurs, guys, right? Obviously, I have told you the cherry red spots on macula will be seen, right? So is something called as the lipid laden macrophages, and there's nothing but the zebra appearance, guys, the zebra bodies appearance. The size of the liver and the spleen both will increase. The zebra bodies appearance would be there, right? If you could just see it over here, guys, that is the zebra body's appearance. The foam cells are there, right? The lipid-laden macrophages are there. Right? So that is something you have to remember. Coming on to the next one, that is the Gaucher's disease. Now you guys already know the Gaucher's disease. How does this occur, guys? How does this occur? Let us just try to understand it in our own way. Then we'll come to the clinical way also. We have Gaucher's disease. Now, guys, Gaucher's disease, basically a case will come, a 3-year-old child. Now, a 3-year-old child presented with abdominal distension. Okay. It presented with anemia, right? Early anemia means the low blood count, the RBC count, abdominal distension, abdomen part is distended, right? There is early bruisability and bone pain. Right? Now, early bruisability and bone pain is there. Just a moment, guys. Don't. So, yes. Now, sorry. So early bruisability and bone pain is there. Now, this is something, no X-ray will be there, the bone marrow will be shown, right? Now, what will happen, guys, over here? Remember that this is called as the Gaucher's disease. Right? Now, this Gaucher's disease, it is the most common lysosomal storage disorder. Right? It is the most common lysosomal storage disorder. Now, what is happening over here, guys? What is the biochemical defect that you have to remember? The biochemical defect. Now, remember that there is something called as glucose cerebrositis. We already know this. Now, glucose cerebrositis will be converting into, yes, ceramide with the help of enzyme beta glucosidase. Okay, or beta glucocerebrositis. Now, imagine this enzyme is deficient. What will happen? There will be increased glucocerebrositis. Right? And this increased glucocerebrositis, or which will be present in non-neural tissue. It is present in non-neural tissue. Now, this is something that you guys have to remember. If it is present in non-neural tissue, remember guys that there is no intellectual disability or there is no mental retardation in these patients. There is no mental retardation. This is deposited in the non-neural tissue. So there is no mental retardation. Mental retardation nowadays, it is called as intellectual disability. It is called as intellectual disability. So remember, it is the child is absolutely normal. I mean, the intellectual is not having intellectual disability. Apart from that, you will also see organomegaly. Organomegaly, guys, right? Now, organomegaly basically means I'm talking about the hepatic and the spleen, the liver and the spleen. So we have this hepatosplenomegaly. So we have no mental retardation and no cherry red spot. No cherry red spot, right? Now what happens, guys? They are giving a picture of the bone marrow, guys. They are giving the picture of the bone marrow, right? Now what happens? Bone marrow, it is the place where all the RBCs, no WBCs, and everything is formed, the platelets, everything, all the bone marrow hematopoiesis takes place. Now imagine a condition where this glucocerebrosidase is getting accumulated in the bone marrow. Now, if there is accumulation of glucocerebrosidase in the bone marrow, what will happen, guys? It causes pancytopenia, right? Decrease in the number of blood counts, right? Decreased thrombocytes. These are another features that you will see. These are the another clinical features that you will encounter. Decreased thrombocytes. Now, if the platelets are decreased, guys, platelets helps in clotting. So if the platelets are decreased, no clotting will take place. These person are more prone to, or it will lead to bleeding. It will lead to bleeding manifestation, right? Bleeding manifestation. Is it clear? So that is something you have to remember. Apart from that, what is the current treatment that we are giving to these patients? What is the current treatment? This RX stands for the treatment, guys. What is the current treatment we are giving to these patients? This is enzyme replacement therapy. Obviously, the enzyme which is not present, just try to give it from outside. We have the enzyme replacement therapy, right? And enzyme replacement therapy, you already know we have one artificial enzyme that you have to know, acid beta glucosidase, right? It is imiglucarase, you can say, right? Glucerase, right? So we have vela glucerase, teleglucerase, right? So we have multiple um, these enzyme replacement therapies. We have substrate reduction therapy, also the second mode of treatment over here, that is the substrate reduction therapy. Or the third important part, since it is causing the change in the bone marrow, the third line of management goes to the bone marrow transplantation. It goes to the bone marrow transplantation. So you just have to remember about this. Let us see what they have given in your Auricle notes. So it is the most common lysosomal storage disorder, enzyme deficient glucocerebrosidase. We have already discussed hepatosplenomegaly, pancytopenias. Yeah, osteoporosis. Yes, the bones will be affected, right? The bones can. That's why the X-ray of the bones will be given. Okay, avascular necrosis of femur, right? Gaucher cells, yes, you can see the lipid-laden macrophages. Basically, they no appear resemble the crumpled tissue paper. You can just see over here, right? The lipid-laden macrophages are there, right? So they appear the like a crumpled tissue paper. You can see over here. Now, and it is treated with recombinant glucocerebrosidase or the enzyme replacement therapy that we have already discussed. Now, coming on to the next one, that is the Tay-Sachs disease. That is the Tay-Sachs disease. Now, guys, you already know. Okay, now coming on to the Tay-Sachs disease, guys. Now, let us first discuss, just try to discuss the Tay-Sachs disease. Tay-Sachs disease. Now, what happens in this disease, guys? Now, imagine a case where a 2-year-old child is present, right? The 2-year-old boy is there. Now, this boy is hypotonic. Basically, the muscle tone is very loose. Okay. Now, it is decerebrate, it is blind. Decerebrate, it is blind. Okay. Very, very important factor. It is blind, right? Fundus examination revealed fundus. We do it for the eyes. You will be studying it in ophthalmology. But as of remember, the fundus examination revealed, yes, the cherry red macular spot. The cherry red macular spot. What is the diagnosis, guys? So this is something which we are talking about, the Tay-Sachs disease, right? So in the Tay-Sachs disease, remember, beta, the enzyme deficient. The enzyme deficient over here, it is, yes, beta hexosa aminidase A. Now, if again there is a question, guys, obviously it is not given over here. If there is an enzyme deficient, that is beta hexosaminidase A and B both, that is called as Sandoz disease. That is called as Sandoz disease. Now, what happens in this Tay-Sachs disease, guys? There is always developmental delay, right? It is always, you will find a developmental delay, right? And yes, it is the neurological deficit that you will encounter in this patient, neurological deficit, right? And yes, and one important factor, that is cherry red spot in the macula of the eye. Cherry red spot in macula. Remember, we have startle reflex. There is increase in the startle reflex, the hyperaction, right? That is something you have to remember. So, but if we talk about the Sandoz disease, these features will be present. But addition to these features, how to differentiate between Tay-Sachs and Sandoz? See, Sandoz is basically the deficiency is beta hexosaminidase A and B both, right? But over here, what are the clinical features? This you will see in Sandoz and Tay-Sachs both. But in addition to this, in Sandoz, you will also see hepatosplenomegaly, Sandoz disease, hepatosplenomegaly, hepatosplenomegaly, right? Plus cardiac abnormality, right? So that is something that you have to remember. Tay-Sachs, all these will be present, but there will be no hepatosplenomegaly, there will be no cardiac abnormality. So that is about the Tay-Sachs disease. Coming on to the next one, that is the Krabbe's disease, guys. Now, Krabbe's disease, let us just try to see over here first what is how this Krabbe's disease patient is presented to us. We have Krabbe's disease. Krabbe's disease. Now, what happens to this case, guys? Remember, a 5-month old child. A 5-month-old child will be presenting with developmental delay. Developmental delay, right? Now, at 9 months, the condition is severely deteriorating. The same child condition is rapidly deteriorating. Rapidly deteriorating, right? Now, at 9 months, you can see that this patient is having opisthotonous, right? Posture with clenched fist. That is being shown in the figure also, which clenched fist. Apart from that, guys, what you will encounter, that is multinucleated globoid cell in the white matter. In the white matter. And one more important thing, that there is no hepatosplenomegaly. No hepatosplenomegaly. Now, this condition, it is nothing but it is the case of, yes, the Krabbe's disease. It is the case of Krabbe's disease. And what is the biochemical defect over here, guys? The biochemical defect. Right? The biochemical defect, and that is galactocerebrosidase will be changing into ceramide. Beta galactocerebroside, right? Now this enzyme will be defective. What will happen? There will be increased galactocerebroside. Increased galactocerebroside. Now, This is something which will result in severe neurological deficits. Severe neurological deficits, right? So that is something you have to remember over here. Apart from that, if you just try to see guys over here, yes, the enzyme defective, it is keratocerebrocytis. They have written peripheral neuropathy will be there, obstruction of the oligodendrocytes, okay, the oligodendrocytes, right? The developmental delay will be there, intellectual disability, mental retardation, along with optic atrophy. Optic basically means refers to the eye, the optic nerve atrophy would be there, right? And the pathology in the cell, in the smear, you will see the globoid cells, right? The globoid cells in the white matter. They are basically the multinucleated globoid cells in the white matter, right? So that is, yeah. Cherry red spots, beta, they can be present, they cannot be present. If you can see over here, these cherry red spots, they are present on the hand, but it is variable. It is variable, guys, right? So again, you can remember the patient will be presenting with febrile episodes. He will come with multiple episodes of fever, right? We have alpha galactosidase A deficiency, burning pain, right? There will be renal failure, guys, and there will be death, right? So that is about the Krabbe's disease, guys. That is about the Krabbe's disease. Now, coming on to one more important part that is called as the ketone body synthesis that I wanted to discuss with you guys, right? So, yes, yes. Now, ketone body synthesis, that is I will be drawing with you apart from this. Yeah, the ketone body synthesis. The ketone body synthesis. Now, guys, you already know in beta oxidation, we have just discussed beta oxidation. In beta oxidation, we have acetyl coenzyme A. We have acetyl coenzyme A. Now, this acetyl coenzyme A, yes, will be like multiple, right? We have multiple acetyl coenzyme A, or I can say there are multiple numbers of this acetyl coenzyme A n times. Now, what it will form? It will form acetyl coenzyme A, right? This is a 4-carbon compound, right? We have multiple, they'll be reacting with each other and it will form acetyl coenzyme A. Acetyl coenzyme A, right? So, yes. How it is formed, guys? Acetyl coenzyme A will be combining with acetyl coenzyme A. Again, you know acetyl coenzyme A is a 2-carbon compound. Acetyl coenzyme A is a 2-carbon compound. Both they will combine with each other and they will form acetyl coenzyme A. Now, what will happen? Yes, there will be again addition of one acetyl coenzyme A over here, one acetyl coenzyme A over here, and the enzyme that is used, that is over here, that is HMG-CoA synthase. HMG-CoA synthase, and the product which will be formed, it is HMG-CoA. Now, what is this HMG, guys? It is hydroxy methyl glutaryl coenzyme, hydroxymethyl glutaryl coenzyme. Now, this HMG-CoA will undergo lysis. It will undergo lysis, guys. It will undergo breakdown with the help of an enzyme HMG-CoA lyase. And what will happen? This acetyl coenzyme A will get removed, right? And again, acetoacetate will be formed. Acetoacetate will be formed. And what are the products? It will form acetone, right? And beta hydroxy butyrate, right? Beta hydroxy butyrate. So you can see this acetone is again a ketone body. Beta hydroxybutyrate is again a ketone body, and acetoacetate is again a ketone body. Now, if I ask you which is the primary ketone body, it is acetoacetate. So let us just try to discuss some questions over here based on ketone synthesis. Now, ketone body synthesis. Now, remember, guys, that this synthesis basically it occurs in, okay, occurs only in liver, right? And which part of the liver, beta? It is inside the mitochondria. Okay, it is inside mitochondria. Mitochondria, that is the mitochondrial matrix. Or I can simply say it is the liver mitochondria. Liver mitochondria. Now, starting material Starting material, it is acetoacetyl coenzyme A, right? If not, it is given. If the option is not given acetoacetyl coenzyme A, just mark acetyl coenzyme A, right? Now, the rate limiting enzyme, over here, that is HMG-CoA synthase, right? So that is the enzyme, right? Common in ketone body synthesis and obviously in cholesterol synthesis also, right? So the ketone bodies which are formed, these are three. We have acetoacetate, acetone, and beta hydroxybutyrate. Now, acetone, it is basically, okay, acetoacetate, it is the primary ketone body. It is formed first. It is the primary ketone body, right? Acetone and these are secondary ketone body. These are secondary ketone body. So that is about the ketone body synthesis. That is about the ketone body synthesis, guys, right? We have already discussed the ketone body synthesis, the Krabbe's disease, right? We have discussed the Tay-Sachs disease, the Gaucher's disease, right? The Niemann's Pick disease, the energetics of the beta oxidation, right? We have discussed the beta oxidation proper, all the steps of the beta oxidation, right? And the concept of beta oxidation. So let's end this today, right? So we have already discussed all this, right? And just you can go to all the links that are being given on the screen for further updates of the classes. Thank you.